reducing sugars are all your monosaccharides. How do we know that something is reducing? So when reducing sugar is added to Benedict's or Fehling solution in which you are having copper ions that are blue in color, they reduce the copper ion Cu2 plus to Cu plus, which converts into red color. So if the Cu plus is converted, if the solution is in red color, we know that this is a reducing sugar because it reduced copper. It gained an electron. So yeah. And glycosidic bond formation now. Okay. Here you can see we are taking maltose, which is glucose plus glucose, or you can take lactose, glucose plus galactose, or your sucrose, which is glucose plus fructose. Your glucose plus glucose, we know glucose, right? Okay. Now here you have your here you have your H, OH, H, OH, here H, here OH, H, OH, H, of course, plus same thing. Okay, here you see this is fourth carbon, this is first carbon. Glycosidic bond happens between first and fourth carbon. Okay, here you are taking OH from this and H from this, and you are basically dehydrating, removing a water molecule. What does this do is basically, I'll just draw like this, and then here you have your other molecule. So this forms O. This is your glycosidic bond. Here you have your H respectively. This is the glycosidic bond. This is 1, 4. So it's 1, 4. I'm sorry, glycosidic bond. Whatever. Okay. As you can see, this is your glucose. First glucose 1, second glucose 4. You're removing H2 molecule, and you get this. This is your glycosidic bond. It's a PYQ, chemical formula of maltose. Just see, maltose is glucose plus glucose. How many does glucose, like you see, glucose is basically C6H12O6 into 2 minus H2O. You get C number won't change. H12 24 minus 2 22, O6 12 minus 1 11. So this is the chemical formula of maltose. Okay, C12H22O11. Now polysaccharides, these are homopolysaccharides. Cellulose and chitin, they are your structural polysaccharides because cellulose you find in cell walls of plants, chitin cell wall of fungi. This is a polymer of beta glucose. This is of NAG, that is N-acetyl glucosamine and glucosamine. This is unbranched. This is also unbranched. Okay, we'll just go through this. Cellulose, it's unbranched cell walls of plants in paper, in cotton fiber. This is not a secondary structure, like this doesn't have any helices. Okay, cellulose, so it cannot hold iron. Okay, just see this. When you are forming, okay, this one, this has helices, you know, this has helices. So let's say a structure, some protein is there, some polysaccharide is there. Okay, a polysaccharide also forms this kind of a structure. Okay, a polysaccharide also forms this kind of a structure. This secondary structure, this helix structure, something. Okay, don't think of it as protein. It has some hollow regions where when you're adding iodine, it contains that. So when you're adding iodine to starch, we know that when you're adding iodine, it turns blue, green, blue, black color, something. We know that, right? Because starch has helix. Helix is present in starch. Whereas in cellulose, when you add iodine, it will not show a blue color because it cannot hold iodine. It doesn't have helices to hold iodine. Okay. Now chitin, it's a polymer of NAG, that is N-acetyl glucosamine. And this glucosamine, it's an amino sugar. It's a chemically modified sugar. It's found in fungal cell wall. It's unbranched, and it's exactly, hence they are unbranched. That's why they don't hold any helices. They don't have any helices. They can't hold iodine. Okay. It's found in, very important, exoskeleton of arthropods, whatever. Cockroach, they have chitin exoskeleton. Hence that's why in a nuclear apocalypse, cockroaches will not die. Cockroaches will be there, of course. Okay. So then you have, these are heteropolymers that are homopolymers that are for storage. Okay, so they are storage polysaccharides. Your starch, it's a polymer of alpha glucose, glycogen, alpha glucose. Inulin is only fructose. This is the only thing mentioned in NCERT. Okay, you can just see here. Inulin is a polymer of fructose. That's the only thing mentioned about inulin. Okay, starch. It's branched. It stores food in plants. It forms secondary helix. Hence holds iodine, which gives blue color. So when you're adding iodine to starch, let's say potato. When you're adding iodine, it gives a blue color because it can hold iodine. It has helices to hold iodine. because it forms secondary structure. And it's storehouse of energy in plants. And okay, I'll explain the structure later. We'll go to glycogen. Same, it's branched alpha glucose, but this is in animals and fungi. Stored food in animal and fungi. Inulin, polymer of fructose. It's stored food in dahlia and asparagus. Okay, now look at this small diagram. Okay, here you have one glucose or something. This is glucose actually. Glucose. When it's normally, this is your linear chain. Okay, when it's unbranched, it's a linear chain. When it's branched, your sixth carbon, that's the carbon that forms the OH from here, forms bond with the other monosaccharide. So this is 1,6 glycosidic bond. At branching, alpha 1,6 glycosidic bond. At normal like linear chain, it's alpha 1,4 glycosidic bond. So that's why you see, this is your linear chain. Here you're having a branch. This is a linear chain, but this is alpha 1,6 glycosidic. This is linear, this is linear. Alpha 1,6. Okay, cool. In a polysaccharide chain, your polysaccharides, whatever. Example, glycogen, you have left end and right end. Glycogen is your alpha glucose. You have left end, you have right end. Your right hand has a free aldehyde, okay, somewhere here. That's what it is. Okay, and this aldehyde, this right end is reducing because it has aldehyde. Left end, it does not have this aldehyde, is used of in glycosidic bond. Hence, it's not reducing. If you want to remember, right, reducing. So your left will become non-reducing. I think, yeah, that's done with polysaccharides and everything. Okay, nucleic acids are your DNA and RNA. Basic issue. Okay, they are biomacromolecules, acid insoluble, pool retentate, more than 10,000 daltons. The building blocks of they are like nucleotides are the building blocks of nucleic acids. Nucleotide is composed of 3 components: phosphate, sugar, and nitrogenous base. Okay, these are the 3 things. When you have only nitrogenous base and sugar, it's called nucleoside. When you have all the 3, so basically nucleotide is your nucleoside plus phosphate. I'll explain better here, don't worry. Let's take only nitrogenous bases here. Okay, nitrogenous bases. These are divided into 2, purine and pyrimidine. Purines have adenine or guanine. And pyrimidine have cytosine and thymine and uracil. Adenine, you see, these have 2 rings. One is a 6-membered ring. Sorry, it's not all carbons. 6-membered. This is 5-membered. 6-membered, 5-membered. These are all only singular 6-membered rings. All are heterocyclic. All have, nitrogen carbon. So it's heterocyclic, not homocyclic. 2 rings, 6 carbon, 5 carbon. Nitrogen, just see the numbering here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Nitrogen is found at 1, 3, 7, 9. So nitrogen on 1, 3, 7, 9. Same for this. Okay, 1, 3, 7, 9. For adenine, guanine, both are present in DNA and RNA. Both are present. Okay. Pyrimidine, you have these 3. Just look at the cytosine. It has 1, 2, 3, 4, 5, 6. At 1 and 3, you find your nitrogen. 1 and 3 nitrogen. 1 and 3 nitrogen. Just look clearly at uracil and thymine. You just see that thymine has the CH3 more than uracil. So it's 5-methyl uracil, thymine. Remember this, very important. These are heterocyclic compounds, single ring, nitrogen at 1 and 3. And cytosine is present in both DNA and RNA. Thymine only in DNA. Uracil in only RNA. That's what. So you're finding these 3, adenine, guanine, and cytosine in all 2. DNA and RNA. This in DNA, this in RNA. These are basically your nitrogenous bases. That's it. Then let's go to sugar. You have, of course, 2 kinds of sugar, your ribose and deoxyribose. That's it, nothing more. Then phosphate. Different structures of phosphate. No, just at different kinds. Like you're substituting everything with H. Just you're removing H. That's your phosphate. Now we are seeing the formation of nucleoside. Nucleoside is your base and your sugar. Your nitrogenous base and your sugar. So let's take here a purine and a deoxyribose. Okay, deoxyribose. Just look at this. I don't want to draw the structures again. Okay, this is your adenine. It has at 9th nitrogen. Remember, 9. And your first carbons, OH. Okay, OH and H are gone. So this is forming. Just look at this. This N is forming like this. You get it right? This is actually called 1,9 N-glycosidic bond because it's nitrogen, not carbon here. Okay. This is for purines. Purines have 1,9 glycosidic bond, 1,9 N-glycosidic bond. Let's take cytosine here. Pyrimidine, which is a pyrimidine. So N, the first nitrogen here. N ka H and your deoxyribose only, or some sugar. Deoxyribose have taken. OH, first carbon OH, and N is first one. So 1,1 dash or 1,1 glycosidic bond, 1,1 N glycosidic bond, or N 1,1, whatever. This is your pyrimidine. So in purines you have 1,9. In pyrimidine you have 1,1. That's the difference. This was the formation of nucleoside, that is your base and sugar. For nucleotide you have nucleoside plus phosphate. So here we have taken, I'll just take this as base. Okay, I write base here, and this is my deoxyribose sugar, and this is the PO4 or what phosphate. You have POH for one, and OH of the sixth carbon, fifth. They are pentose sugar. Sorry, pentose fifth. Yeah, okay. Your OH from here and H from here. H2 removed in everything. It's a dehydration process. Okay, and yeah, this CH2 is connected with the O here. So P O minus double bond O. One O is connected. This you can see is an ester bond. You see, or if you're taking this O here and O minus man, it's an ester bond, but it's not a diester. Don't forget, phosphoester bond. That's it. And this is your normal base. Same. Phosphate plus nitrogenous base plus sugar is nucleotide, which is nucleoside plus phosphate. Let's take adenylate acid, which is given in NCERT. So just look at this. Adenine, your base. This I've taken ribose here because NCERT I've taken ribose. And you have your phosphate group. If you want, we can see it in NCERT. Yeah, I think we better understand here. Okay. We have adenine, purine. Just remember, 1379. You have nitrogen in purines. In pyrimidines, 1,3. That's it. We have pyrimidine, yeah, 1,3. Uracil. 5-methyl-uracil is thymine. Okay, this I'll explain again. Adenylic acid. You have your adenine, which is nitrogenous base. Your ribose sugar, OH is there, that's why. And your phosphate group. This is adenylic acid, which is a nucleotide. This is basically, you have your ribose sugar plus adenine, it's adenosine. This is uracil plus your ribose sugar, uridine. Okay, your pyrimidines become denes in nucleosides, and your purines become scenes in nucleosides. So adenosine, guanosine, thymidine, uridine, cytidine. Okay, that's what I'll show in the next one. Okay, nitrogenous base: adenine, guanine, thymine, cytosine, uracil. Nucleoside: adenosine, guanosine, thymidine, cytidine, uridine. Just don't get confused like scene. Of course, cytosine, we know that it's a pyrimidine, so the nucleoside will be cytidine. And uridine, of course. Nucleotide: adenylic acid, guanylic acid, thymidilic acid, cytidilic acid, uridylic acid. Now, why did I tell phosphoester bond here? Because the bond, this is phosphoester, there's no double diester. The phosphodiester bond is formed between two nucleotides. So you have one nucleotide here and another here. Okay, I'll just quickly show this. You have your phosphate group, CH2. This is your nitrogen, sugar and base. This I've taken deoxy again. And the third carbon. Third carbon's OH group with the OH group of phosphate. Phosphate is connected to the fifth carbon, if you know. So fifth carbon's phosphate's OH group with third carbon's OH group are connected, like and then the dehydration, removal of H2O, OH of this and H of this are removed, and this forms here a single bond O. You see. So that's why this is a diester. You see, this O is there in this. This is there, like P. You see, okay. This is a diester because this both O's are required. This can't be just for this. You are using. Okay, you understand. Basically, why phosphodiester is that you have. Just look at this, and look at this. You need this O for this to form with this, and this O to form with this. Okay, and this is 3 and this is 5. So 5 dash 3 dash 5 phosphodiester bond. That's it. And look at this. This is NCERT slide. First nucleotides, sugars. Third carbon's OH forms phosphodiester bond with fifth carbon of another nucleotide, sugar. Fifth carbon का nucleotide no, sugar no. इसका nucleotide phosphate का से you're forming basically. And now I go to enzymes. I'll explain enzymes from NCERT. I'll just keep turning these pages so that nothing feels like we are missed. We are just revising a little. Biomolecules, just that relative abundance of carbon and hydrogen with respect to other elements is higher in our living organisms than in Earth's crust. Cl3CCOH, this is the trichloroacetic acid that is used. Then you have, okay, biomolecules are all the carbon compounds that we get from living tissues. Then amino acids are organic compounds containing amino group, acidic group, and the R group if they are both on the same carbon, alpha carbon, and they are called alpha amino acids, substituted methanes. We saw these tables. This is the intracellular ion that's abundant. This is extracellular ion. We know this. This is the Zwitter ion, CO minus NH3, and this is the H. You see. I don't think we have missed anything. Lecithin is phospholipid, myelin sheath. This is the sphingomyelin that you find. Okay, nucleosides, uracil. Okay, here you see, here you'll understand. Now, adenosine and uridine nucleosides. Yeah. And here just one of the PYQs I guess was like they gave adenosine or guanine and adenyllic acid and some other, and they asked which of these is a nucleotide. Of course, adenyllic acid. DNA, RNA function as genetic material. This yeah, these are your secondary metabolites. You have pigments. Okay, yeah, pigments, polymeric substances. And then you have, yeah, this is your abundance thing. Proteins, then nucleic acid, then carbohydrates, lipids, and inorganic ions. What is the most? 70 to 90. Proteins 10 to 15. This is 5 to 7, 3%, 2%, 1%. Protein is always a heteropolymer and never a homopolymer. Essential as should be supplied through our diet. You remember all these, right? Like, okay. The amino acids like alanine, cysteine. This is sulfur containing. Proline is just mentioned. Tryptophan is aromatic amino acid. Lysine is the basic amino acid. This is neutral amino acid. This one, proteins carry out many functions in living organisms. Some transport nutrients across cell membranes, are integrated proteins or transmembrane proteins. Some fight infectious organisms, antibodies, hormones, enzymes. Enzymes we'll learn now. And yeah, okay. Collagen is most abundant in animal world, rubisco and biosphere. Cellulose is homopolymer of beta glucose. Starch is for alpha glucose. Inulin is polymer of fructose. Right end is reducing, left end is non-reducing. This one, I think we already kept enough emphasis. Starch forms helical secondary structures. In fact, starch can hold I2 molecules in the helices. Cellulose does not contain complex helices and hence cannot hold. This is what they showed, the branching here, and this is the normal polymer. You can see here. The 1,6 glycosidic bond. This is the 1,4 glycosidic bond. And yeah, these are aminochemically modified. Complex one. This is a complex polysaccharide. Polysaccharides are mostly homopolymers. That's what we. And nucleic acids. Yeah, everything is covered in this. Proteins, we know left hand is first. Yep. Okay, these are the structures of proteins. 1 degree, that is primary, that is linear. Helices and pleated. Tertiary, you have the hollow things here, and in the quaternary, disulfide bonds and more complex. Like here, just look at this. Quaternary, tertiary structure is absolutely necessary for many biological activities of proteins, that is enzymes. Enzymes have tertiary structures, and quaternary is basically the manner with the spheres. Okay, you know this has different linear spheres like plates and all. All of those are quaternary. Many folds. Okay, adult human hemoglobin consists of 4 subunits. 2 are identical to each other. Hence 2 subunits of alpha type and 2 subunits of beta type together constitute hemoglobin. And then we have peptide bonds. Okay, they explain here. Peptide dehydration. This is third carbon. This is what we discussed. Nucleic acids exhibit a wide variety of secondary structures. For example, okay, the secondary structures exhibited by DNA is the famous Watson and Crick model. We find like this. This one. They are actually anti-parallel. You see, if this 5' is going like this, here you always get your 3'. I think it's mentioned somewhere. Yeah, it's anti-parallel. I'll read this. This model says that DNA exists as a double helix. Okay, we actually learned this in molecular basis of inheritance even better. But yeah, the 2 strands of polynucleotides are anti-parallel. They run in opposite direction. The backbone is formed by the sugar phosphate sugar chain. Nitrogen bases are projected more or less perpendicular to this backbone. Okay, we have AG. A का complement is T and G का complement is C. Your adenine, thymine, guanine and cytosine. Okay, that's exactly. This you can see like A sub. Okay, I don't think this is clear in my book, but it's fine. Okay, there are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. Guanine and cytosine and adenine and thymine have 2 hydrogen bonds, 3. Each strand appears like a helical staircase. Each step of ascent is represented by a pair of bases like AT, CG, GC, TA. So it's a pair of bases. At each step of the ascent, the strand turns 36 degrees. So one full turn of the helical strand would involve 10 steps or 10 base pairs. Okay, so 360 degrees, one full turn. Okay, when drawing it, okay, this is all not important. You see that BDNA, this is called beta DNA, beta helix, so BDNA. Now, this dynamic state of body constituents.